In today's lesson, we're going to be covering the energy, enzyme, and biological reaction. Right, let's begin. Um, okay. Energy, we know that energy cannot be measured or weighed directly. It can be detected only through its effect on matters, which is including its ability to move object against opposing forces such as friction, gravity, pressure, or to push chemical reactions toward the completion. When we talk about energy, we know that energy can exist in different states. Two forms of energy is kinetic energy and potential energy. Kinetic energy is putting in motion, is the energy of an object because of its motion. For example, if you hit a baseball or you see a falling rock, this is an example of a kinetic energy. Then we have potential energy. Potential energy is stored energy, that is the energy an object has because of its location or the chemical structure. A boulder on top of hill has potential energy because of its positions in Earth's gravitational field. In biological system, potential energy can be released in a chemical reaction and it is called a chemical energy. Chemical energy is stored in bonds between the atoms. We can convert potential energy into the kinetic energy, or we can convert kinetic energy into the potential energy. Then we have laws of thermodynamics, which describes the energy flow in natural system. Transformation of energy is the thermodynamic. When discussing thermodynamics, we know scientists refer a system which is an object under the study. A system is whatever we define it to be, a single molecule, cell, planet, or anything. Everything outside the system is its surrounding. Three systems we must know is isolated system, closed system, and open system. Isolated system does not exchange matter or energy with its surrounding. A perfectly insulated thermos flask is an example of an isolated system. Then we have closed system. Closed system can exchange energy but not matter with its surrounding. Earth is a closed system. It takes in a great amount of energy from sun and releases heat. But essentially no matter is exchanged between Earth and rest of the universe. So it is a closed system. Then we have open system. Open system can exchange both energy and matters with its surrounding. For example, all living organisms are the open system. Let's look at the diagram. So first diagram is representing isolated system. It shows that does not exchange matter or energy with its surrounding. Then we have closed system, exchanging energy with its surrounding but not matters. In an open system, both the exchange of energy and matter is happening. So these are the systems in thermodynamics. First law of thermodynamics addresses the energy content of system and their surrounding. First law of thermodynamics states that the energy can be transformed from one form to another, or transferred from one place to another, but it cannot be created or destroyed. That is, in any process that involves an energy change, the amount of energy in a system and its surrounding remain constant. This law is also called the principle of conservation of energy. Must remember this law. How does the principle of conservation of energy apply to biochemical change? Molecules, we know, both have kinetic and potential energy. Kinetic energy for molecule above the absolute zero is reflected in constant motion of molecule, whereas in the potential energy for molecule is chemical energy, which is energy contained in arrangement of atoms and chemical bond. The energy content of reacting system provides part of information required to predict the likelihood and direction of chemical reaction. For chemical reaction in which a molecule is broken down to a simple molecule, Energy content of the reactant is larger than the energy content of the product. Thus, energy usually progresses to a state in which the product have the minimum energy content. When this happens, the difference in the energy content between reactant and product in the reacting system is released to the surrounding. Then we have the second law of thermodynamics, which considers changes in the degree of order in reacting system. Second law of thermodynamics states that this tendency towards disorder formally in any process in which a system changes from an initial to a final state, the total disorder of a system and its surrounding always increases. In thermodynamics, disorder is called entropy. If the system and its surrounding are defined as entire universe, the second law means that as changes occurs anywhere in the universe, the total disorder or the entropy of the universe increases constantly. As the first law of thermodynamics asserts, however, the total energy in the universe does not change. Okay, so we looked at two laws of thermodynamics. First law states the law of conservation of energy, which says that energy cannot be created or destroyed but can be changed from one form to another. Then we look at the second law, which is law of entropy. When energy is changed from one form to another, there is a loss of unstable or energy, or waste energy goes to increase the disorder. Then we have free energy and spontaneous reaction. Applying the first and second law of thermodynamics together allow us to predict whether any particular chemical or physical reactions will occur without an input of energy. Such reactions are called spontaneous reaction in thermodynamics. So we know that spontaneous reactions does not require input of any energy. Two factors related to the first and second law of thermodynamics must be taken into account to determine whether a reaction is spontaneous or not. This is number one is change in energy content of the system and change in its entropy. Reaction tend to be spontaneous if the product have less potential energy than the reactant. The potential energy is system in its enthalpy, N mean to put into, enthalpy and mean to heat, symbolized by H. Reaction that releases energies are called exothermic reaction, where products have less potential energy than the reactant. Reactions that absorb energy are called endothermic, where the product have more potential energy than the reactant. Second factor is reaction tends to be spontaneous when the products are less ordered or more random than the reactant. Reaction tend to occur spontaneously if the entropy of the product is greater than the entropy of the reactant. Change in free energy indicate whether a reaction is spontaneous. The portion of a system's energy available to do work is called free energy, which is symbolized by G. The change in free energy, pronounced as delta, can be calculated for any chemical reaction from the following formula. So delta G equals delta H minus T delta S. Delta H is the change in enthalpy, T is the absolute temperature in degrees Kelvin, and delta S is the change in entropy. For a reaction to be spontaneous, delta G must be negative. As the above formula tells us, both the entropy and enthalpy of a reaction can be influenced by the overall delta G. We can say that the free energy is influenced by entropy or enthalpy. In some processes such as combustion of methane, large loss of potential energy, negative enthalpy dominates in making a reaction spontaneous. In other reactions such as melting of ice at room temperature, a decrease in order dominates. Once we know the delta, what the free energy or change in free energy is for a reaction, we can determine if the reaction will proceed spontaneously. Okay, spontaneous reaction typically reaches an equilibrium point rather than going to a completion. At the equilibrium point, we must remember both reactant and products are present, and the reaction typically are reversible. They both are in the equilibrium, a balanced state between opposing factor pushing the reactant in the either reaction. Let's look at the diagram. The equilibrium point of a reaction, no matter what a quantities of glucose 1-phosphate and glucose 6-phosphate are dissolved in water, when equilibrium is reached, there is 95% of glucose 6-phosphate products and 5% of glucose 1-phosphate reactant. At the equilibrium, number of reactant molecules being converted to the product molecule equals the number of product molecule being converted back to the reactant molecule. The reaction at the equilibrium point is reversible. It may be made to run towards the right by adding more reactant or towards the left by adding more product. For the reaction whose delta G or free energy is near to the zero, they are readily reversible by adjusting the concentration of products and reactants slightly. Reversible reactions are written with reversible reactions are written with a double arrow um, as shown in the diagram. A plus B is reactant and C plus D is the product, and they are represented with double arrow, which mean they are reversible reaction. We can change reactant to product and product back to the reactant. Metabolic pathway consists of exergonic and endergonic reaction. An exergonic reaction releases free energy, so the delta G is negative because the product contains less free energy than the reactant. In an endergonic reaction, the product contains more free energy than the reactant, so the delta G is positive. Let's look at the diagram. Figure A shows the exergonic reaction. Free energy is released, product have less free energy than the reactant, and the reaction proceeds spontaneously. Then figure B is endergonic reaction. Free energy is gained, product have more energy than reactant, and the reaction is not spontaneous. In metabolism, individual reactions tend to be a part of a metabolic pathway. In one type of metabolic pathway, which is also called catabolic pathway, kata means downward, as in the sense of rock releasing energy as it rolls down a hill. Chemical energy is released by breakdown of a complex molecule to a simpler compound. An individual reaction from which chemical energy is released is the catabolic reaction. Another type of reaction is called the anabolic reaction. Ana means upward, as in sense of using energy to push a rock up a hill. In an anabolic reaction, it is an individual reaction that requires a chemical energy input is called the anabolic or biosynthetic reaction. In catabolic, we are breaking things. In anabolic reaction, we are making things. The overall delta G of a catabolic pathway is negative, whereas the overall delta G of anabolic pathway is positive. Then we have adenosine triphosphate, the energy currency of the cell. ATP hydrolysis releases free energy. The breakdown of ATP is a hydrolysis reaction and the result in formation of a adenosine diphosphate and a molecule of an inorganic phosphate. ATP can be hydrolyzed further to adenosine monophosphate AMP, but this releases somewhat less energy than the hydrolysis of ATP to ADP. Phosphate group from the ATP hydrolysis coupled reaction. The hydrolysis of ATP in water in a test tube is an exergonic reaction that releases free energy that simply warms up the water. How do living cells couple the hydrolysis of ATP to an endergonic reaction so that energy is not simply wasted as heat? The answer is the energy coupling. The use of exergonic reaction, ATP hydrolysis, to drive an endergonic reaction. In other words, two reaction becomes coupled reaction. In a coupled reaction, ATP is hydrolyzed and its terminal phosphate group is transferred to the reactant molecule of the endergonic reaction. The addition of a phosphate group to a molecule is called phosphorylation and the modified molecule is said to be phosphorylated. Phosphorylation makes a molecule less stable and more reactive than when it is unphosphorylated. In the figure A, we can see that chemical structure of AMP, ADP and ATP. There is a phosphate group, there is a ribose sugar and a adenine. With one phosphate group, the molecule is known as AMP. With two phosphate, the molecule is known as ADP. Each added phosphate packs additional potential and chemical energy into the molecular structure. The figure B represents the hydrolysis reaction removing a phosphate group from the ATP. Okay, the figure on the right represents energy coupling using ATP in synthesis of glutamine from glutamic acid and ammonia. Figure A, without ATP, reaction is not spontaneous because delta G is positive. So glutamic acid reacts with ammonia to form the glutamine. ATP hydrolysis is an exergonic reaction. Coupled with ATP hydrolysis, the glutamine synthesis reaction is spontaneous because the net delta G is negative. In step 1, C, the free energy of terminal phosphate of ATP is transferred to glutamic acid. The complex reacts spontaneously with ammonia to produce glutamine and delta G is negative. Coupling reactions occurs in living organisms consuming a tremendous amount of ATP. Cells also couple reaction to regenerate ATP. In exergonic reaction, supply energy for ATP exergonic reaction supply energy for ATP production, ATP synthesis, and endergonic reaction, which is energy requiring reaction. In endergonic reaction fueled by energy from the ATP hydrolysis. ATP hydrolysis, which is an exergonic reaction, it is an energy releasing reaction. ATP is a renewable resource that is synthesized by recombining ADP and phosphate. Although ATP hydrolysis is an exergonic reaction, ATP synthesis from ADP and phosphate is an energy requiring endergonic reaction. The energy for ATP synthesis comes from exergonic reaction that involves breakdown of complex molecules that contain an abundance of free energy in form of carbohydrates, proteins and fat in food. The continual hydrolysis and resynthesis of ATP is called the ATP-ADP cycle or coupled reaction. Role of enzymes in biological reaction. Activation energy represents a kinetic barrier for a reaction. This initial energy investment required to start a reaction is called the activation energy, which is symbolized by EA. Molecule that gains the necessary activation energy occupies what is called the transition state, where bonds are unstable and are ready to be broken. In the diagram figure A, you can see the activation energy barrier in oxidation of a glucose. So this is the reactant, products, energy released by reaction, and the activation energy. We said that the activation energy is the minimum amount of energy that is required to activate atoms or molecules to a condition in which they can undergo chemical transformation or physical transport. When writing activation energy, it is always measured in joules per mole or kilojoules per mole. Heat commonly provides the energy needed for a reactant molecule to get to a transition state, and therefore to speed up the rate of reaction in biology. Using the heat to speed up a reaction is problematic for two reasons. Number one, high energy or high temperature destroys the structural component of cell, particularly proteins, and an increase in temperature would speed up all the possible reaction in a cell and not just the specific reaction that are the part of metabolism. Enzyme accelerate reaction by reducing the activation energy. How can the rate of reaction increase without a raise in temperature? By the use of catalyst, a chemical agent that accelerates the rate of reaction without itself being changed by the reaction. The process of accelerating a reaction with a catalyst is called the catalysis, and we say that the chemical agent responsible catalyzes the reaction. The most common biological catalysts are the proteins called enzymes. Enzymes increase the rate of reaction by lowering this barrier, that is, by lowering the activation energy of the reaction. Because of the rate of a reaction is proportional to number of reactant molecule that can acquire enough energy to get to the transition state. Enzymes make it possible for greater proportion of reaction molecules to attain the activation energy. Do's and don'ts of enzymes include: number one, by lowering the activation energy, enzymes do speed up the rate of spontaneous or exergonic reaction. Enzymes do not supply free energy to a reaction. Therefore, enzymes cannot make an endergonic reaction proceed spontaneously. ATP hydrolysis can be coupled to an endergonic reaction to make it proceed spontaneously, but not alone. Enzymes do not change the delta G of a reaction. In the figure 6.9, you can see effect of an enzyme in reducing the activation energy. The reduction allows biological reaction to proceed rapidly at relatively low temperature that can be tolerated by the living organism. Enzymes combine with reactant and are released unchanged. Figure 6.10, combination of an enzyme hexokinase in blue and with a substrate glucose in yellow. Hexokinase catalyzes the phosphorylation of glucose to form glucose 6-phosphate. The phosphate group that is transferred to glucose is not shown. Note how the enzyme undergo a conformational change, closing the active side more tightly as it binds to the substrate. What is a substrate? The reactant on which an enzyme acts is called the substrate, or substrate if the enzyme binds two or more molecules. Active site is the place on enzyme where catalysis occurs. The active site is usually a pocket or a groove that is formed when the enzyme protein folds into a functional three-dimensional shape. Then we talk about the cofactor. A cofactor is a non-protein group that binds precisely to an enzyme for catalytic activity. Role of a cofactor in an enzyme catalytic activity varies with cofactor and the enzyme. Some cofactors are metallic ion, including iron, copper, magnesium, zinc, and manganese. Other factors are small organic molecules called coenzyme, which are often derived from the vitamins. Some coenzyme bind loosely to the enzyme. Others, called prosthetic groups, bind tightly. Enzyme reduces the activation energy by stabilizing the transitional state. Enzymes stabilize transitional state doing so through three major mechanisms. Sorry. Number one is enzyme bring the reacting molecule together. Reacting molecule can assume the transition state only when they collide. Binding of reactant molecule to an enzyme active site brings them close together in correct orientation for catalysis to take place. Enzyme expose the reactant molecule to altered changed environment that promote catalysis. In some system, the active site of enzymes contain ionic group whose positive or negative charge alters the substrate in a way that favors catalysis. Enzyme changes the shape of substrate molecule as mentioned previously. The active site can distort substrate molecule into the conformation that mimics the transitional state. Let's see a diagram representing the catalytic cycle of enzyme. Step 1. The substrate bind to enzyme active site forming an enzyme substrate complex. Number 2. Enzyme catalyzes the reaction, breaking a bond in substrate molecule to generate two products which are then released from the enzyme. Number 3. Enzyme active site can bind another substrate molecule and catalyze the reaction. Conditions and factors that affect the enzyme activity. Enzyme and substrate concentration influence the rate of catalysis. Presence of excess substrate, that is, at a high concentration, the rate of catalysis is proportional to the amount of enzyme, and enzyme concentration increases the rate of product formation. As the enzyme concentration increases, the rate of product formation also increases. As the substrate concentration increases, the reaction rate initially increases as the enzyme and substrate molecule collide more frequently. However, as the enzyme molecule approach the maximum rate at which they can combine with reactant and release product, increasing substrate concentration has a smaller and smaller effect, and the rate of the reaction eventually levels off. Here in the figure 6.12, effects of increasing enzyme concentration A or substrate concentration B on the rate of an enzyme catalyzed reaction is given. Figure A, rate of reaction as function of enzyme concentration, substrate at a high concentration. And figure B represent the rate of reaction as a function of substrate concentration. Enzyme amount is constant in this. Cell adjust enzyme activity to meet their need. Several mechanisms are used in the regulation, which include the competitive and non-competitive inhibitors, a form of non-competitive control called allosteric regulation, and number 3 is the covalent modification of enzyme structure by addition or removal of the chemical group. Competitive and non-competitive inhibition. In competitive inhibition, an inhibitor competes with normal substrate for binding to an enzyme's active site. That is, Competitive inhibitors have shape resembling the normal structure closely enough to fit into and occupy the active site, thereby blocking the access for normal substrate and slowing the rate of reaction. If the concentration of the inhibitor is high enough, the reaction may stop completely. Competitive inhibitors are useful in enzymes research because their structures help identify the region of a normal substrate that binds to an enzyme. Then we have non-competitive inhibition. In non-competitive inhibition, an inhibitor binds to an enzyme at a site other than its active site and changes the conformation of enzyme so that the ability of active site to bind substrate efficiently is reduced. Inhibitors, either competitive or non-competitive, differs with respect to how strongly they bind to an enzyme. In reversible inhibition, the binding of an inhibitor to enzyme is weak, and when the inhibitor releases, enzyme activity returns to the normal. In irreversible inhibition, an inhibitor binds so strongly to an enzyme through the formation of a covalent bond that the enzyme is disabled completely. Looking at the figure 6.13, how competitive and non-competitive inhibitor reduces enzyme activity. Figure A, competitive inhibitor molecule resemble the substrate and competes for active site. Substrate is unable to bind when inhibitor is bound to the active site. In non-competitive inhibition, non-competitive inhibitor bind at a site other than the active site, causing enzyme shape to change so that the substrate cannot bind to the active site. Okay, then we have allosteric regulation. In mechanism of allosteric regulation, allo mean different and stereo mean shape. Enzyme activity is controlled by the reversible binding of regulatory molecule to the allosteric site. A location on the enzyme outside the active site, the mechanism may either increase or decrease enzyme activity. Because allosteric regulatory molecules work by binding to the site separate from the active site, their action is non-competitive. In one conformation called the high affinity state, the active form, the enzyme binds strongly to its substrate. In other conformations, the low affinity state is the inactive form. The enzyme binds the substrate weakly or not at all. Binding with regulatory substance may induce either state. Binding an allosteric activator converts it from the low to high affinity state and therefore increases the enzyme activity, and binding an allosteric inhibitor converts an allosteric enzyme from high to low affinity state and therefore it will decrease the enzyme activity. Regulation of a type in which the product of a reaction acts as a regulator of the reaction is termed as the feedback inhibition, also called the end product inhibition. Feedback inhibition prevents cellular resources from being wasted in synthesis of molecules made at the intermediate steps of the pathway. Regulation of chemical modification. Many key enzymes are regulated by chemical linkage to other substances, typically ions, functional group, and a phosphate or methyl group or unit derived from the nucleotide. The regulatory substances induces folding change in enzyme that increases or decreases its activity. pH and temperature are key factors affecting the enzyme's activity. Effect of pH. Typically, each enzyme has an optimal pH where it operates at peak efficiency in speeding the rate of biochemical reaction. On either side of the pH optimum, the rate of catalyzed reaction decreases. The effect become more extreme at pH value farther from the optimum until the rate drops to zero. An enzyme's dependence on pH typically is caused by the ionizable amino acids. A change in pH from the optimal value alters the charges of those amino acids, which modifies the conformation of the protein and eventually causes the denaturation of the enzyme. Then we have effects of temperature change. Effect of temperature changes on enzyme activity reflects two distinct processes. Number one, temperature has a general effect on chemical reaction of all kind. As the temperature rises, the rate of chemical reaction typically increases. This effect reflects increases in kinetic motion of all molecules with more frequent and stronger collisions as the temperature rises. Number two, temperature has an effect on all proteins, including enzymes. As the temperature rises, the kinetic motion of the amino acid chain of an enzyme increases, along with strength and frequency of collision between enzyme and surrounding molecules. At some point, these disturbances become strong enough to denature the enzyme. Hydrogen bonds and other forces that maintain its 3D structure break, making the enzyme unfold and loses its function. RNA-based biological catalyst, ribose. RNA-based catalysts, or known as ribozymes, are the part of biochemical machinery of all the cells. Most of the known ribozymes speed the cutting and splicing reaction that remove the surplus fragments from RNA molecule as part of their conversion into the finished form. Ribozymes can act as both as enzymes and informational molecule when cellular life first appear. The earliest form of life therefore might have inhabited an RNA world in which neither DNA nor protein played critical role. If so, ribozymes, the most recently discovered biological catalyst, may have existed for the long time. This brings us to the end of chapter number 6. From this chapter, um, you must remember laws of thermodynamics, difference between kinetic energy and potential energy, then the factors that determine if the reaction is spontaneous or not. Just the description of the factors, not details. What is the difference between exergonic and endergonic reaction? How ATP hydrolysis release free energy, and what is activation energy? Do's and don'ts of enzymes, the role of enzyme in chemical reaction, and what are the three mechanisms that stabilizes by which enzyme stabilizes the transition state? These are the three key points, must remember them. Difference between competitive and non-competitive inhibition, what is allosteric regulation, and then what is the end product inhibition or the feedback inhibition, effects of pH and temperature change. Yeah, that's all from this chapter, and do revise the key concept, review key concepts. Thank you.